But genomic medicine is a vast topic. And today I want to give you a flavor of how these technologies are becoming embedded in practice in the UK. Actually, in England in particular, because the devolved nations, Scotland, Northern Ireland, and Wales are taking a slightly different approach. But I think it'd be useful for you to see strategy. I will give some clinical examples. I hope there'll be something here for everyone. I'm going to keep it at quite a high level because I mentioned before the topic is vast. The reality of the practice and the integration of genomic technologies into the care pathways is very complex. There are still regional variations, and of course, there are variations internationally. But I'm really giving a very sort of England centric approach. This might have a slightly different flavor from other sessions you've had. Again, I'm grateful to Professor Akandi for giving me some insight into the sorts of topics that will be of interest to this audience. My background is in informatics and cancer, but I don't want to talk too much about the specifics and the minutiae. And if you're not familiar with some of the technology, although I understand that there are people from the labs here who might be interested in some more technical aspects, I've tried to deal in a flavor of applications, some of the challenges and strengths of these approaches. I've put in a couple of pauses where I'm going to ask questions because there are interesting discussion points, and I'm really, really interested to hear the thoughts of the audience. I've given my email address, so please put some questions in the chat as we go through. There will be a couple of points where I will specifically ask for your views. If anyone wants to send me an email to follow up on anything, I'd be delighted to hear from you. So I hope there's something here for everyone. Again, it is quite high level. I have put some clinical examples in, but again, it may be slightly different from the case report sessions you've had historically. So now I need to get my slides to advance. Okay, so of course, the fact that some diseases have a heritable component has been known for a very long time. But it was only with the rediscovery of Mendel's work in the early 20th century that scientists were able to get to grips with the inheritance of diseases. And fairly quickly, it was recognized that some human diseases followed a monogenic Mendelian inheritance pattern. Now, not going to talk too much about genetics today, but the key thing is genomic medicine has been promising a lot for a long time, and it's only now got to the point where the technology is allowing us to deliver. So very quickly after de Vries and others rediscovered Mendel's work, Archibald Garrow, working with Bateson in Cambridge, he recognized that alkaptonuria was inherited according to a Mendelian pattern. And he rationalized that it resulted from the loss of function of a key metabolic enzyme. In this case, it was homogeneous dioxygenase. And Archibald Garrow actually coined the term inborn errors of metabolism. So these metabolic disorders are really there at the start of modern genetics, as the modern genetics really began in the early 1900s. And then, of course, throughout the 50s, with the discovery of the sequence of DNA, and then the development of techniques that allowed us to manipulate DNA and work out the DNA sequence, that genetic or genomic medicine began to, or perhaps overpromise. There were a number of cancer targets identified quite early on, which turned out not to be druggable, for example. So I've been in this game for quite a long time. And I was been a little bit disappointed that the translation of advances in genetics have taken some time to improve patient outcomes. But in the last 10 years, the technology has really advanced at a rate that we can now begin to do testing and understand the genetics of disease at a population level that allows us to offer the best care to our patients and offer meaningful, helpful, actionable advice. So, which is really the key. So today I'm going to talk about how these technologies are beginning to transform the management of a range of different conditions. So by way of a very recent history, the genomics revolution, for want of a better term, was really kick-started around about 2013 when David Cameron, who was then the British Prime Minister, who had a child who suffered from a rare disease, he became aware that the technology to determine an entire individual's genome was at a point where it was becoming affordable. And you may have seen talks like this before, where people have shown graphs of the declining cost of DNA sequencing. But now, pick a figure, maybe $1,000, you can determine the genetic code of an individual's entire genome, the whole list of G's, A's, T's, and C's that specifies every gene, protein, and ultimately phenotype. So when it became affordable to sequence genomes at scale, the model changed. Instead of trying to work out how how disease was inherited, we could ask different questions, or we could ask the question in a different way. I'm going to give some examples of this. But it was realized if you were able to look at the genomes of people in a population, you could learn a lot more about the association between variation and disease. So, at a very simple level, if you've got a large cohort, within it, you may have individuals with a particular disease, and within that subgroup, there'll be certain variants which are overrepresented. And it gives you a different way of getting a handle on the genetics of disease. And I don't want to say too much about this. And I'm really going to focus on monogenic disease, which is the change in function of one gene having a measurable output, a phenotypic effect. Of course, these monogenic disorders tend to be quite rare. So inborn errors of metabolism, they are rare at a population level. Of course, the impact on the sufferer is enormous. More common disorders, cardiovascular disease, obesity, type 2 diabetes, they have a much more complex genetic footprint. There are a number of different genes, different variants in genes, all have a small contribution to the disease. I'm not going to talk about that today. So the 100,000 Genomes Project, it was really a prototype. Was it possible to gather a large cohort of patients, take detailed histories, collect rich demographic and clinical data on these individuals, sequence their genomes, and then begin to pull out actionable intelligence? So when the 100,000 Genomes Project was designed, it really had two arms. One was to look at rare disease, and another was to look at cancer. I'm going to mention cancer in a moment, but thinking about rare disease, I guess questions were asked. Why look at rare? Why not look at common diseases? Well, actually, if you gather together everyone in the UK with a rare disease, you're looking at about 3.5 million people. So if you have a different paradigm for understanding the basis of those disorders, you've got the potential to treat a lot of individuals. And because historically rare disorders are difficult to diagnose and treat, you're potentially offering a diagnosis and the mechanisms to screen family members for a lot of individuals who may have been waiting years for a diagnosis. You may have heard about the diagnostic odyssey, where someone with a rare disorder may wait 5 to 8 years or go through at least 5 years of fairly exhaustive investigations trying to find a diagnosis. And that diagnosis in itself might not necessarily lead to treatment, but psychologically, having a diagnosis helps. And again, it may be treatable. There may be routes for screening family members. So the 100,000 Genomes Project was really a proof of principle. Can sequencing be done at scale? And then, what sorts of things can we do with the information? How can that then inform what a genomic medicine service might look like? So the proof of principle that gathering lots of genomes from lots of individuals, you can begin to pull out variations associated with disease, provided that that prototype or the kind of testing the NHS might offer. And there are a couple of aspects to this. So I'm going to mention this later on. And again, I won't say too much because I want to throw some questions back to the room. But you need infrastructure to make sure that someone with a genetic disease gets the right treatment at the right time, make sure they get referred into the system and then managed afterwards. There have to be defined criteria for testing, again, just so that the testing is more focused and so that the intelligence gathered from the testing is actionable. I'm going to talk about that. And another important thing, and this is kind of where I came in. So Professor Candy mentioned earlier on, my background is in informatic and genomic research. It's really important that those in the health service understand what can be done, what is the state of the art. Because these are new technologies, there are many interventions which are now possible. But if clinicians, doctors, the healthcare team are not aware of them, then patients aren't going to be getting the best service. And you end up with things like regional, national, and international disparities because of access to these services have to be as equitable as possible. And genomic medicine is, or education in genomic medicine is at the heart of this. So Genomics England Limited, through Genomics Education, they've got lots of resources if you want to go and get little bite-sized learning chunks. In Cambridge, I'm responsible for the master's program in genetic medicine, which largely brings doctors up to speed in the state of the art. What can we do? How do we do it? And what are the implications of it? So I think it's useful just to think about how genomic services are organized in the UK. Now, the NHS, or NHS England, has published a series of strategy documents. I've hyperlinked some of these documents. I've made a PDF available for those who want it, if you want to go have a look at these documents. So the NHS articulated a very clear strategy where they wanted to put genomics and information technology at the heart of patient care. And I won't go through all of this diagram, but this explains how the different teams work together. So if a patient is referred on, and the referral may have come from primary care, it could have come from pediatrics, cardiology, whatever. If there are a series of criteria that the patient needs to fit before they undergo testing, because if they don't fit the criteria, then the test won't necessarily be informative. I'll give an example of this in a moment. But so if someone presents to a specialist with good evidence that they have a genetic disease and the diagnosis, screening for the underlying genetics is going to be of value, then we have centralized testing hubs where the DNA is analyzed. And again, there are a whole raft of different technologies, which I'm not going to talk about, different high throughput means of either looking at a small panel of genes or looking at the whole genome. And again, these are all described in the appropriate guidance documents that I'll come on to. But basically, if an individual meets the criteria for testing, the testing will be done with very high standards in a regional center, and then that test will be interpreted and then returned to the clinician who will then manage that case from there on. Now, other teams, I mean, clinical genomics, clinical genetics, have teams of genetic counselors because getting a diagnosis of a genetic disease is going to be life-changing. So it has to be presented appropriately. If you're collecting lots of complex information, that in itself is a valuable resource. So patients who undergo screening are consented to make their data available to the National Genomic Research Library. So I'm going to come back to this later on. So we've got a huge bank of genetic information, which is coupled with rich patient data, which is a really, really powerful resource for scientists to go back and mine to look for new insights. And then, of course, you need the clinical leadership as well to make sure that the latest innovation in genomic medicine is recognized in the service. But I'm going to give an example of this. I mean, this may seem a little bit complex. This is where we kind of are at the moment. But I'm going to talk through a couple of examples of this, and it'll hopefully make sense how someone with a genetic disease is managed in the UK. But we've developed infrastructure, and this shows how England has been carved up into 7 different regions. I think, for example, anyone in this region are going to be referred on to services in Oxford. Those in the southeast are going to be, again, I think regional services are actually based in London. But anyone asking for a genetic test in one of these regions, the testing will be done in a regional hub so that the quality of the analysis, the analysis and the results returned have to meet very stringent clinical standards. So this carving up of the UK into these 7 regional centres allows for consistency, and hopefully it's removing regional variation. So anyone who presents with a genetic disorder anywhere in the UK who meets the criteria should get the same level of service. Again, this is evolving very rapidly, but this is kind of where we are. I was going to throw a question over. So, Professor Candy, if you can take the temperature of the room. At the moment, we could theoretically, it's almost cost effective now for us to screen everyone's DNA, but we don't do that. I mean, can anyone think of a good reason for doing it and think, no, but now that we can screen someone's entire genome, why aren't we doing that routinely? So again, if I can throw that over to the floor, anyone have any thoughts on what some of the barriers might be to really effective genetic screening? I hope I haven't sent
Participants, if you
everyone to
could
sleep.
just, yeah, so I was just going to say, if you could just pop your answers in the Q&A box, or if you would like to raise your hand, that would be great, and I can unmute you. So we'll just give a couple of seconds for that. Thank you. You see, we've got one answer, Dr. Kenneth.
Okay.
We've got one answer, and it says it's too expensive.
So that's a really good point. So cost is absolutely a concern, but even if we could afford it, we're still not at a place where if we have somebody's whole genome, we can actually do something with that information. So we end up throwing a lot of information away. So really, that's a good answer. And it's one reason for not doing it. But we're getting to a point where it will become affordable. But then is the analysis using that information in a way which is meaningful to the patient, we're still some way off achieving that. So if you think in the UK at the moment, I think we have 3 nationwide screens for breast cancer, we've got colorectal cancer, we've got cervical cancer. And again, these aren't necessarily genomic screens, but we're still in a position where we can't offer screening more broadly because the number of individuals who'll benefit from an early diagnosis will be outweighed by those who may receive uh, warning that they contain potentially pathogenic alleles, but we don't really know if it's going to cause disease in their lifetime. So the key thing is we need to be much more focused because otherwise we don't know what to do with the information. And that's really the key. So for that reason, at the moment, rather than screening everyone, other than the cost, screening will only be offered, genetic testing will be offered if there's a strong family history of disease. And I'll give an example of that in a moment. There may be a child who's developed difficulties from birth, who hasn't received a diagnosis. I'm going to give an example of this as well. There may be people who already have. They may have a genetic condition they know about, in which case it's really just advice, or they may need confirmation of the variation in the genes involved. And a really big component of this is family planning. If an individual with a genetic disorder or who has a genetic disease in the family, if they're making decisions about having a family, they want to know what are the risks of passing on to their children, and is there testing available, or for someone who's already pregnant, is there a prenatal test which will be able to identify a genetic disease? So the key, if an individual, there has to be strong evidence that someone has a genetic disease before they will be referred on for further screening. And if a GP, for example, a primary care physician, meets a patient who feels they have a genetic disease, and the GP is satisfied that there's probably this good cause, and I'll give an example of that in a moment, they will refer on to clinical genomic services. They will give the individual a form to fill in, and they will then get as much information as possible about the disease in that individual's family. So I mentioned before, there are a number of test directories that very explicitly describe the conditions under which a test will be offered. There's a directory for rare disease. There's a separate document for cancer, and this is an extract from the eligibility criteria for rare disease testing, and I've pulled out the range of metabolic disorders or suspected metabolic disorders. If individuals meet the criteria, they can then be put forward for testing. If you think about lysosomal storage disorders, there are a range of lysosomal storage disorders. I won't go through this in detail, but if you look in the test directory for rare disease or the eligibility criteria, it'll say what, for which presentations is there strong evidence to allow that individual to go forward for testing. It says which specialties, because again, there are gatekeepers in the UK. A GP, a primary care physician, can only order one or two tests. They would refer on to clinical genetics or the metabolic disease clinic or the or pediatrics as appropriate. So only individual specialties can request tests, and then they will become responsible for managing that case eitherwise. So if you're someone with a suspected lysosomal storage disorder, of which there are quite a few, the directory will describe the circumstances under which a test is appropriate, and then it also talks about what test will be performed. There are a number of genes that could potentially be involved in lysosomal storage disorders, and in this case, like I won't talk too much about the technology. Whole exome sequencing will be done, which is where only the coding, the DNA in the coding parts of the genome will be determined, or they will look at a panel of genes, just a targeted set of genes which are known to be informative. So what it means is you're not generating a lot of information you can't do anything with. You're asking some really specific questions, which in most cases are going to be meaningful for that patient. For some disorders, there might be a single gene. With cystic fibrosis, for example, sickle cell disease, you would only be put forward for testing in that one particular gene. In the case, I mean, there are a range of lysosomal storage disorders. I just pulled out some of the genes that are tested. I mean, you see hexosaminidase A in there, which is involved in Tay-Sachs disease, for example. So this screen is quite a broad one. It's looking for genes which are known to be, there's very good evidence that variations in these genes will underlie the specific disorder. So again, it's making it as targeted and as specific as possible. In the presentation, I put some links to these documents if you want to have a look at them. So I'm going to switch gears for a moment, and the next couple of slides, I'm going to talk about cancer. I realize that might be of less interest to this group. It is a subject I'm more familiar with, but I think it illustrates is an example of a referral pathway. and very specific steps and very specific criteria that have to be met before genomic testing is done, and hopefully it'll make sense. So we have here a little patient vignette. 43-year-old woman presents to her GP because her mother's been diagnosed at a young age with breast cancer. She then becomes aware that a number of other female relatives have had an early diagnosis of either breast or ovarian cancer. And, with stories of people like Angelina Jolie in the news, there's increased awareness that there are familiar cancers and that there are tests for susceptibility to breast cancer, for example. So what would happen in this case is a GP will take a fairly basic family history, and if they're satisfied that the criteria are met, that there's a strong evidence that this is genetic, then the patient will then be referred on to the local clinical genetics service. So what will happen then, I think I mentioned this earlier on, they will be sent a family history questionnaire. So basically they'll be checked again to make sure that there's strong evidence that there's a genetic cause for this cancer. And this will be this information will be gathered by either a clinical geneticist, a consultant clinical geneticist, or a genetic counselor. They'll put together a pedigree. And again, I won't go through the pedigree here, but this is a very classic BRCA pedigree with breast and ovarian cancer, near female relatives, diagnosed at an early age. And the proband here, the patient, is indicated with an arrow there. So the important thing is that this patient is asymptomatic. So I'd like to throw this back to the floor. So you've got a situation here where you've got an individual with strong family history of cancer who's asymptomatic, has asked for a genetic screen. Does anyone have any insight into what happens next and who would be offered screening? So again, I'll give you a couple. This is quite an important point, and I'll give you a minute or two. And if maybe Roxanne, if any answers go into the chat, you can let me know. But basically, what would happen next? Anyone have any thoughts?
Thank you. I think participants can go to the chat box or the Q&A box. Got a lot of people now, about 40 participants, so we should be more interactive now.
Excellent. I know it's a little bit difficult via Teams. I wish I was in the room with everyone. It'd be much easier.
So we'll just give some seconds to get responses.
Any thoughts?
Not yet. Okay, maybe we can go on.
Okay, I'll move on. So I guess the important thing is here that the woman who presented would not be tested. She would not have her genome sequenced, which would sort of be the obvious thing to do. And the reason is there's a huge amount of diversity. And I'm going to come back onto that in a moment. So what would actually be done is a relative that they would seek permission to do a DNA test on an affected relative. So they wouldn't screen the woman unless there was no material available from someone who was affected. Because what could happen is the woman could be tested and they might not find anything, and which just causes more confusion. But if a BRCA mutation is found, which is known to cause a loss of function in an affected individual, you'd be much more confident that that variation was causative, that was pathogenic. So it's only once a causative variant is found in an affected family member that the woman who presented to her GP would be screened for that variant. And then her siblings, other family members who wanted to be screened, would be screened as well. So again, I realize it's difficult to do this really interactively. And again, I'm managing to fill the time. So rather than throw this out to the floor, I'll answer this question. So if it transpires that the woman who presented to her GP has a causative mutation, has a susceptibility mutation in the BRCA1 allele, which will give her a 60 to 80% chance of developing cancer in her lifetime. Now, what can we do about it? The reason for testing is getting intelligence. We can do something which is going to make the patient's life better. So the obvious thing to do is increased surveillance. I'm trying to remember, is it 50 that breast cancer screening starts in the UK? So there'll be earlier screening with various imaging techniques to pick up a potential cancer early. Some, if they've got a high risk mutation, because some mutations carry a greater risk than others, might think about preventive surgery, whether that's prophylactic, mastectomy, which is a really, that's a really critical decision to make, and it's got to be informed by the best information possible. There may be an option for chemo prevention, perhaps taking tamoxifen prophylactically. Might be family planning decisions. If the woman's of reproductive age, does she want to have children? Does she want to have screening? She's got a chance of passing this mutation on. This diagnosis is life-changing. So even though the individual may have as I say, a 60 to 80% lifetime chance of developing cancer, dealing with that is an enormous challenge. The individual has to be supported. And there may be options for some lifestyle modifications which could potentially reduce the risk. And of course, there are implications of a negative test as well. You might have a situation where some sisters are screened, and then one of them turns out not to be carrying mutation, not to be at risk. And that in itself can be very challenging. And it's not uncommon for genetic counselors to see individuals who get a negative test who are not at risk of cancer, them experiencing survivor's guilt. So again, got to get these decisions right. And hopefully I'm making the point that when you do screen someone, you want to make sure that the insights developed from that test are as meaningful and as actionable as possible. So moving on to rare disease now. So again, a rare disease may only affect a few hundred. Any individual rare disease may only affect 100 or so people worldwide. But if you group rare disease together, millions of people are affected, millions of people who are unsure, millions of people waiting for a diagnosis. So again, this has a huge effect on an individual and for a younger individual's families as well. I mentioned earlier on the 100,000 Genome Project focused on rare disease and cancer. So again, a little case study of a little girl called Jessica. And soon after birth, she began to experience fits. There were problems with developmental delay. She had problems with movement. And she underwent lots of investigations, MRIs, EEGs, some more invasive investigations like lumbar punctures, but she wasn't able to get a diagnosis. So for a family, this adds to the stress enormously. And I talked about the diagnostic odyssey, where historically it can take more than 5 years for someone with a rare disease to get a diagnosis. And again, it might not be able to help treatment, but it will help the individual manage the disease. Again, I was going to throw this question out to the floor, but I'll just answer it here because I want to leave some time for questions at the end. So if you're enrolled in 100,000 Genomes Project, in addition to sequencing the DNA of the affected individual, it's also important to sequence the DNA of that individual, the affected individual's biological parents. And the reason for that is the amount of variation between individuals, human populations, is much, much, much larger than we realized before these large-scale, cohort-wide genome sequencing initiatives began to take place, which launched in 1990. It looked at the genomes of 4 or 5 individuals, and that cost, so it cost $3 billion. And then but that drove the technology that brought down the cost of sequencing. Using the traditional Sanger sequencing approach, which is a very accurate and exhaustive method, I think it costs about £10 million to sequence a genome using Sanger sequencing. Around about 2007, there was a new generation, next generation, or now generation sequencing, whatever you want to call it, really increased the throughput and decreased the cost. And once we started sequencing lots of genomes, we realized there was much more variation between 2 unrelated individuals than we expected. And if you take 2 unrelated people, sequence their genomes and compare them, you might find differences. You will find differences in more than 5 million places. So if you're trying to identify a change, a variant, which is directly related, to use that old cliche, you're looking for a needle in a haystack. So you need to narrow that down. And in with every generation, the germ cells will generate about something like 100 different mutations, or this might occur early embryogenesis. So between closely related individuals, between parent and offspring, you're looking at maybe 100 differences. So if you've got an unaffected parent and an affected child, you've got a much more targeted series of changes to look at to work out if they're potentially causative. So that was the real power of the 100,000 Genomes Project. It was looking at trios, looking at families, because it also allows you to look at the zygosity is the change, is homozygous for how the disease is manifested, if at all. So this is really the big challenge now in genomics, is taking someone's DNA and trying to work out if there's a pathological change in there which underlies the disease. And so in many cases now, we tend to focus on those variations for which we've got good evidence. I'm going to come back to that in a moment. So in this case, we have in a databases, we have a reference genome, a standard genome that any new DNA sequence can be compared to. So we've got big clusters of computers that can crunch through this data and do these massive comparisons. So if you compare Jessica to the reference genome, if you look at the 4 million differences. And of course, we think, well, this is where machine learning or AI comes in. It can crunch through and try and predict which of these changes could cause a disease phenotype. Actually, that doesn't work very well. If you ran computational analysis on the variations in Jessica's genome, the software predicted that there'd be about 3,000 protein changes, and they could be all different kinds of protein changes, but they predicted these variations could have lots of effects. And of course, most of them don't. So there were actually only 67 differences between Jessica and her parents that narrows it down. And then the way that they narrowed it down to the causative variant was by comparing each of these in turn, which is now a much more tractable exercise, against a reference of known variations. And that's stored that's curated in a resource called PanelApp. And PanelApp is growing all the time, and it uses a traffic light system. So we're identifying variations in human DNA all the time. But if we don't have evidence that that change is pathogenic, it'll be flagged up in red, and it's not used for interpretation. And they'll be working their way through the system. Investigators will be looking at trying to determine if that change could potentially. It could be based on population studies. They may actually take the protein, introduce that variation, see what it does. Because it's really, really hard work. So there are lots of changes. We don't know what they do. There are some changes, we might have some evidence, but again, the evidence is developing that that change in the DNA is absolutely causative for a disorder. And then what PanLab uses, and this is what is used in most interpretation, are variants that we know, we're absolutely certain that that change in the DNA is going to cause this phenotype. So in this case, when the informatician, the bioinformatician, who's the specialist who crunches the data, when they looked at these 67 changes, they found there was really good evidence that change, a loss of function in one gene, in this case is a gene involved in glucose transport, this could explain all of Jessica's symptoms. So there's really good evidence this gene would lead to the fitting, the developmental delay, so the motor study. So this is basically, it was found out that Jessica had a form of a rare form of a GLUT1 deficiency syndrome. So Jessica was able to get a diagnosis, and this is one of the early successes of the Human Genome Project. And I hope this illustrates some of the principles and some of the challenges in going from DNA to a diagnosis. So in this case, Jessica lost one copy of the SLC2A1 gene. Version of this protein is an important glucose transporter. So it's involved in transporting glucose because the blood-brain barrier. So loss of function, of course, is going to have a number of neurological sequelae. And in this case, by giving Jessica a low carbohydrate or ketogenic diet, they were able to manage the fitting. So this, again, this is a good example of someone who waited years for a diagnosis, got a diagnosis, and was able. And I'm sure these figures are changing now, but I think it was reckoned that from the original 100,000 Genomes Project, that about 25 to 30% of participants got something usable from it. It's not necessarily a treatment, but at the very least, they knew the mutation so that that could be used in screening family and cascade screening for family members, or in prenatal in family planning decisions as well. But increasingly, finding what the mutation is, and using that information to help manage the patient's disease. And again, coming back to the earlier point about why we don't screen everyone, well, there still is a fairly modest number of genetic changes for which that we're really confident that they're associated with the disease and we can do something with it and it's going to be of value to the patient. Now, I'm going to say a little bit about newborn screening in the UK and where that's going for here. So in the UK, there's a simple heel prick test, some blood is taken, and there's a fairly traditional panel of biochemical tests. Obviously, look for common disorders, relatively common, sickle cell, cystic fibrosis, congenital hypothyroidism, and also a range of metabolic disorders, of inborn errors of metabolism, for which early, obviously, dietary interventions can help, manage the stop the disease sort of manifesting itself. So there are fairly modest panel of disorders for which newborn screening is performed in the UK. So an initiative which is being driven by Genomics England is called the Generation Study, where they're going to sequence the entire genomes of 100,000 babies. But rather than try and identify every single potential metabolic disease, they're focusing on 200 conditions which are treatable. So the idea is to expand the repertoire metabolic disorders or disorders could manifest themselves early on with potential developmental effects for which there's some treatment. Again, going in parallel with this is the research data, it's all banked. And it means you can go back and start asking some different questions. And I'm going to give an example of that, how we can use all of this banked data to ask some really interesting, really important questions. And the other thing is, what the 100,000 Genomes Project created was an infrastructure for gathering cohorts of patients, for consenting them, and then for storing the data in, and then for creating an environment in which that data can be accessed and then mined for key insights. So in some ways, so there will be a number of children for whom this screening will provide an early diagnosis and it will be useful. But actually, again, this is a proof of principle which will hopefully inform newborn screening going forward. So the last in infectious disease, and to be absolutely honest, infectious disease is not a topic with which I'm intimately familiar. But of course, during the COVID-19 pandemic, everyone became very aware of these huge endeavors to sequence large numbers of COVID genomes. And so if you sequence large numbers of genomes, then allows you to trace the evolution of potentially more pathogenic strains very, very quickly. So for the genomics, or for epidemiologists interested in monitoring the genomes of infectious diseases, then these high throughput technologies allow evaluated very quickly. But I'm not going to talk about that. Again, that's a little bit outside my wheelhouse. But I will talk about how sequencing patients' DNA helped to enhance outcomes with individuals suffering from COVID-19. So I'm sure, as most of you are aware, there are two broad biological mechanisms that led to a higher risk of mortality in those infected with COVID-19. So there was susceptibility to infection in the first place. So the more susceptible you were to infection, the more chance was of a poor outcome. And then, of course, there were a group of individuals who had a hypersensitive reaction and ended up with severe lung damage, ended up being ventilated, and again, very high mortality. The response to inflammatory signals and the susceptibility to infection, these two components, which were important in trying to identify those at risk of a poor outcome. So quite quickly, the 100,000 Genomes projects and others pivoted their original purpose to start looking at the genomes of individuals who became critically ill. So they gathered genomes of a number of individuals in ITUs throughout the UK. So this was UK-wide this time, and not just England. So the idea was, if you can look at the genetics of a large group of those who get critically ill, can you identify genetic variants that put those individuals at risk? And will this give any insights into targets? Are there any processes that we could improve outcomes? So the way this was done with a genome-wide association study with which you might be familiar. So again, this isn't used routinely in individuals for diagnosis. This is done at a cohort level. Historically, it was done prospectively. You get a large cohort of individuals with a disease, a large cohort of individuals who don't have the disease. You would do some DNA, you do some genotyping, and try and work out which changes were more common in the disease group. But now, with all of this data gathering we've been doing, our databases are now full of complete genomic sequence data from a diverse group of individuals. And now you can you can pick a specific defined outcome. You can split your cohorts into those with and without that outcome. And it could be a discrete effect, like the presence or absence of disease or response to a drug, or it could be continuous, like blood pressure or body weight. And that's traits, because those have a number of genes, which variants in which are associated with those outcomes. But just thinking about more binary, the presence or absence of a variation that causes a very specific measurable effect. So this experiment was done effectively using a material that was already available. And so the genotyping was performed. Like I won't talk about the technology too much. A number of different ways you can look at a genotype. You can use single nucleotide polymorphism arrays that allow you to identify the presence or absence of a single change at a number of positions throughout the genome. You might miss quite a few things there, but if you have enough SNPs, you've got pretty good coverage, and you'd be quite confident that some of your SNPs will be quite close to the causative gene. And then they also did some whole genome sequencing as well to basically confirm the SNP arrays. So they're able to identify a group of variations in, and then they compared that to a reference population. In this case, they used samples that were drawn down from the UK Biobank. There was a lot of quality control steps, which I won't talk about because different populations aren't necessarily homogeneous, and you need to control for that. So there was a lot of very sophisticated statistics used just to make sure that you don't get false positive results. But at a very simple level, you've got two groups of individuals. There are a group of individuals, in this case, you've got a couple of thousand individuals who were critically ill in ITU. And I think in this case, for every affected or every ill individual, they looked at 5 normal controls. So they compared that to 10,000 individuals who didn't have COVID. And they looked at every single locus they could to see come up more often than you'd expect by chance, where certain genetic changes overrepresented in the defined population, in this case, the critically ill. So with some very sophisticated statistical analysis, I've linked the paper for those of you who want to go and look at it, because again, it lays out very beautifully how to do a systematic, methodical genome-wide association study on a fairly heterologous population, and do some statistics that make you quite confident that the associations that you're finding are real. So I think once it would run through a couple of rounds of quality control, I think there were 8 regions in the DNA with which changes were seen more often than in the critically ill group of controls. So mechanistically, these changes made sense. So they found a cluster of genes in chromosome 12, which are involved in antiviral responses. So again, if you have loss of function here, these individuals are more susceptible to becoming infected by a virus. And it's been known for a long time that susceptibility to infectious disease has a heritable component, and they were really able to narrow down mutations or variations in which genes made you more susceptible to infection. And these are involved in the interferon pathway. And also they found the variation in interferon receptor put people at an enhanced risk of in genes. There was dipeptidyl peptidase 9 and the tyrosine kinase 2 receptor. These are involved in inflammatory responses. So with this huge amount of number crunching, this huge amount of analysis, the simple message was the individuals who had high levels of this the tyrosine kinase 2, and these individuals, if you have high levels of tyrosine kinase 2, you're more likely to have this increased inflammatory response to the fibrosis and pulmonary damage. If you had low expression of the interferon receptor, you're at higher risk of being infected in the first place. So there are actually inhibitors of this family of proteins. That's called was it baricitinib of this tyrosine kinase, which will actually suppress the inflammatory responses and therefore can be used as a treatment for COVID. So again, it's a really nice example of how we can, at a population level, look for variations which are associated with a defined outcome, and then we can then use that for a test, not only to identify individuals at risk, but also identify potential therapeutic targets. Let's check how I'm doing for time. So I think I'm about to finish up. So just a few very general conclusions before I take some questions. So at the moment, there's no point doing a genetic test. Some actionable intelligence. And so the Genomic Medicine Service in the UK is based on offering tests which are going to inform treatment. And I mean, there was a little bit of angst. A clinical geneticist might have a patient in front of them who, in their opinion, very clearly has a genetic disease, but don't meet the criteria and won't be tested. But these panels are revised all the time, and there are cases can be made for exceptional testing. But the rules around testing are quite closely prescribed, and again, it's to stop, I guess, people having tests that aren't going to be meaningful. And the other, I alluded to this earlier on, this banking of data is really, really useful. I know that people are anxious about the genetic information. Lots of ethics in place. The data is highly anonymized. If you try and analyze this data, you've got to do it inside a protected environment, privileged environment. You can't take the data out. Anything which could potentially identify the patient is removed, which can sometimes make things a challenge, but that's, I think we have to protect the anonymity of the people in the database. So the bottom line is, if you've got a lot of DNA information and a lot of patient information, you can do a lot. There's an astonishing amount that can be done in silico, computational analysis that can grind through the variation and try and identify what changes predispose people to defined outcomes. So I think the most positive mention is if we can integrate genomic medicine more effectively into the mainstream, it really is going to deliver truly individualized or personalized care. I guess the most important thing is disease prevention. If we identify people at risk and intervene early, then potentially we can we can prevent huge amount of illness. And think about that on the healthcare system. It can provide diagnosis. So someone with a rare disease can provide diagnosis. In cancer, for example, it might allow for a very precise diagnosis to be made and allow the identification of specific mutations which are targetable. So again, that's the key. It's the prevention of disease, diagnosis of disease, and then giving insights into the most effective management individualized to that patient. So I think I'm going to stop at that point, and thank you for your attention.